Welcome to Redefining Medicine, an intimate and personalized program that illustrates a different side of the practice of medicine. Our in-depth conversations will focus on mentors and motivators who are consistently reshaping, redefining, and rediscovering the field of medical healthcare. I would like to welcome Dr. Valter Longo. Director of the Longevity Institute of the University of Southern California, Dr. Valter Longo is a world-renowned expert on the benefits of intermittent fasting and creator of the nutritional breakthrough, Fasting Mimicking Diet. Thank you so much for joining us today, Dr. Longo. I greatly appreciate your time. Let's go back and see if you can reflect back to the time when, in your youth, when you were back in Italy, and what initially sparked your interest in the sciences?
In Italy, actually, nothing sparked my interest because I was a musician. And all I did was music, and that was the reason also to come to the US when I was 16. But my interest in science really started in my second year in college. I was a music major in Texas, and for whatever reason, I didn't want to continue with the music. And I really thought that it was incredible to me that nobody was working on aging. And I mean, I didn't know anybody was working on aging. And so both from a science point of view, but also from a medical point of view, I thought, why don't we work on aging? It seems that all diseases come in old age, and or at least the major ones. And so that was it. And I thought that the biochemistry training was going to be which is going to be best for what I wanted to do. And so that's what I did. Both the undergraduate and my PhD at UCLA was in biochemistry. But I also got a lot of medical training. I spent a couple of years in the pathology department and in the medical school at UCLA. So I really had an opportunity to, to get the training that I wanted. So good choice, I think.
What instrument did you play?
Guitar. Yeah.
Do you still play?
I still play a little bit.
Keeps you young?
Yeah, I still try to, mostly recording, studio recording.
Wow.
But so I try to at least keep the minimum going.
So what was the impetus to go into studying aging? Was there something in your personal life or family history, or just something out of thin air that came and said nobody's working on aging? What was it that fascinated you about that?
In the book that I just published, my suspicion is that I was in the room when my grandfather died. Usually kids are kept out of the room, when somebody dies. For some reason, I was in there. And, I think that probably, when you're 6 years old and you see somebody dying that you were very close to, I'm assuming it's stuck in my head, for in a very major way.
Right.
And then probably, 15 or 13, 14 years later, I probably, and he died fairly early. He was not, he died of something minor that turned out to be a major problem. He had an ulcer that then got worse and eventually killed him. So, maybe I realized that without really paying attention too much to it. And then when I had the first opportunity, instead I thought music, or I could figure out how people age and die. And so I thought that there was no competition there. I mean, even though I love music, but so I think that's probably the main factor. Although I never thought about that. It's not like I was thinking, my grandfather died, I saw it, and this must be the reason, but I suspect that's what it was.
So after you finished your PhD and you've now moved on to USC, I believe, and when did your research start in how fasting helps the body, and what have you found?
No, this started much earlier. So this started, in 1992, when I got to UCLA, Roy Walford was the world leading figure for aging and nutrition, and he was the pioneer of something called calorie restriction. And I joined his lab, even though he was in pathology and not in biochemistry. And so that, of course, started my research on restriction. But then I really got very frustrated with this comparative biology. Take an old mouse, take a young mouse, compare, or an old person. I mean, Roy was doing, at the time, he was doing an experiment on himself and other 8 people. It was the first calorie restriction human study in Arizona in a place called Biosphere 2. And so I met Roy actually through teleconference because he was locked up in this place in Arizona. So I thought this is great, really pioneering. But if we want to really turn medicine around with aging as a focus, treat aging, we need to know the molecular biology. And so I left after 2 years and really not having much accomplished, not because of Roy, but because it was just too premature to try to figure out what calorie restriction did without the genes. So I went back to the biochemistry department, and immediately I started working with starving bacteria and starving yeast. And of course, having been in Walford's lab, it made sense. It was almost like natural for me. Let's see what happens when they starve. And sure enough, I just will switch bacteria to water or to medium with no nutrients in it, and buffer with no nutrients in it, and they will live a lot longer. They will be resistant to stress, and they would be much healthier. And then, most of my work then was with yeast, a unicellular eukaryote, same thing. And then I thought, these have been separated by hundreds and hundreds of millions of years. How could it be that they respond to starvation by being healthier, longer lived, stronger? And that's when I think I started, then devoting all my time to this. And then, of course, but the first thing was genes. So what makes them live longer when I starve them? So first.
How long were you starving them for?
Permanently.
Wow.
Yeah, this is permanent starvation. I mean, of course, they can do it because they're microorganisms that are set up to do that. They have more reserves inside than what it takes to have a full lifespan. And so then we started with the genes, and the question was, well, okay, what are the nutrients that matter? And what are the genes that mediate this? And that was 1994, and I remember, in 1994, within 6 months of going back to the biochemistry department, I had worked out the sugar pathway, and this was the first pathway ever identified, the first genes, by the way, and the first pathway to ever identify to control aging in any organism, right? And but I had designed a new model, a new way of doing it. It was too many new things, And even my boss would say, Valter, you're just 5 years ahead of where you should be because nobody's going to believe any of this. And sure enough, just nobody paid attention to it. And but I mean, I knew that this was real. And so, and this sugar, I figured out controlled something called RAS and then something called PKA. And then together they control these transcription factors, which then control a lot of genes, which control aging. And so almost a complete pathway. And it would take a few years or a number of years for that to be published. In 2001, eventually we published that. So it took 7 years to get that out there in a major science publication.
How was it received at that time?
Well, I mean, it originally got rejected over and over and over, So we sent it to Cell, one of the major journals, scientific journals, and they just didn't believe the system, didn't believe any of it, and so that was tough. But we kept going, and in 2001 it just exploded because then I also made the case even before in the 90s that I thought this was all conserved, So these genes that control aging were not just controlling aging in yeast. I said the same genes I think control aging in all organisms, And of course, at the time, it was laughable, and people thought, come on, yeast. Now you're telling us from yeast data that this is conserved. But sure enough, 10 years later, we and many others then confirmed both in 2001, the science paper actually presented both the amino acid, the protein, and the sugar pathways, which we had, published on in some form before, and and the TORP TOR acid kinase pathway, which we identified went on to be now what's recognized as the most powerful pro-aging pathway in all organisms. So I think luckily we still managed to, from something very negative, come up with something, a double discovery in 2001 in Science, one of the world's leading journals. So in the end, it turned out to be a good story. Luckily I didn't give up.
Now, is this the initial research that helped to propel the FMD, the Fasting Mimicking Diet, or was there research that came after that helped bring that diet to life?
No, of course, it's all built on step by step. So once we had the genes, then of course we started moving to mice and other organisms, and started doing similar research on genetics of aging in mice, and even in humans. And then eventually, I think around 15 years ago, I started thinking. I knew the results of the calorie restriction world. I knew the calorie restriction had incredible effects on diseases. It could knock down. We didn't know at the time, but certainly we suspected that that it could do that. And eventually, the monkey studies demonstrated that a number of years later. But we also knew from the work that Weinberg was also a student of Roy Walford. Weinberg had gone to University of Wisconsin, and he was the leading leader of this monkey study, and eventually showed that monkeys, about 60% of them develop on this Western diet. They develop diabetes. 0 of the calorie-restricted monkeys did, and the cancer and cardiovascular disease were reduced by 50%. And these are monkeys, are not mice. So pretty clear that this, and also if you combine that with the Walford human study, which shown remarkable effects on risk factors for diseases, you could tell the calorie restriction had the potential to revolutionize medicine. There's no doubt. The problem was that it didn't really extend the lifespan of monkeys very much. So it had these incredible effects on diseases, but the monkeys died about at the same time, right? And that was really the big push for the fasting mimicking diet. It's like, okay, I think now, because we have so much molecular data, we're a little bit ahead of everybody else, that we can figure this out first. And that's where I thought, what if you just do it once in a while? Could you eliminate all the problems of calorie restriction, and now eliminate the burden of calorie restriction? Of course, nobody wants to be calorie restricted and be anorexic, basically anorexic. Walford, when he came out of Biosphere 2, it looked like a skeleton, somebody that came out of Auschwitz. You knew the power of this because his blood pressure was 90/55 or something like that, and cholesterol was down to 100. But at the same time, you look at him and the other guys and thought, this is not the way to do it. And so what if you just do it once in a while for a few days? Could this have long-lasting effects? And could this just eliminate the burden? Because you could tell people, maybe you could do it every 3 or 4 months. So that's where the fasting diet started. But it started first for cancer patients, or it started also for cancer. But I think there was 2 parallel things. One was what I just said, and the other one was we knew that if you start the yeast, they become very protected against chemicals, all kinds of toxins that you can throw against it. But then we also knew that the proto-oncogene, so what in cancer is always active as an oncogene, reversed this protection. So then it occurred to me about 12, 13 years ago, and I remember calling a colleague and said, what, I think I figured out a way to separate all cancer cells from all normal cells. And I remember she said, yeah, whatever. And but it was true, meaning that if you starve a system and it's normal, it knows exactly what to do, whether it's a yeast, a bacteria, or a human cell. It'll go into this starvation response mode, and it sits there until food comes around again. But a cancer cell, because it has an oncogene in it, it will rebel by definition. It's not even something. That's why I knew it had to be right, because that was not my definition of cancer cells, it was everybody else's definition of cancer cells. And I thought, is it possible that nobody's ever thought about this, you know? And so by definition, it will not respond to starvation, right? It will continue. So I say, perfect. You know, you give an order, all the good cells protect, all the cancer cells keep on going. And sure enough, it worked beautifully. I mean, even better than we thought. So you starve a mouse, you give them high-dose chemotherapy, nothing, no side effects. You do it with a controlled diet, all dead. You know, so we saw some incredible results of all alive, all dead, which you almost never see in biology. So we knew that this was a very good thing, and you know, so now we have a number of clinical trials running for fasting mimicking diet and chemotherapy, but also in novel therapies, not just chemotherapy, but kinase inhibitors, immunotherapy, hormone therapy. So eventually, I think we're going to do this combination, fasting mimicking diet with all kinds of therapies.
On all cancers or a select few?
We have seen very few cancers that don't respond. You know, again, it's a fundamental property, and I know for people it's hard to believe, but this fundamental property is of all cancers, right?
At what stage?
Yeah, we believe at all stages, and in fact, even in the prevention level, right? So we showed that if you take mice at middle age and you start the FMD, the tumor incidence will be cut in about half, and many of the tumors will be postponed and benign versus malignant. So you cut it in half, postpone this half, and then benign versus malignant at the prevention level. So it seems to work in prevention, and also particularly in combination with chemotherapy with all kinds of cancers. We've seen very few exceptions. And
how many days of the fasting were you doing?
The cancer, the human cancer fasting mimicking diet is 4 days long. We're now also came up with a 5-day long higher calorie version for patients that may have lost weight or that tend to lose weight. And so it's 3 days before chemo, 1 day after. And of course now we're going to try to combine it with all kinds of different interventions, you know, kinase inhibitor, immunotherapy, etc. So, we have a number of trials, I think 5 clinical trials ongoing right now, and we expect at least another 5 in the next year they're going to start. I mean, of course, a lot of hospitals and universities from around the world are interested. So now we have MD Anderson, Mayo Clinic, USC, University of Leiden, the Oncology National Cancer Institute in Milan, et cetera, et cetera. You know, so some of the studies in a year are going to be done. If everything goes well in a year, I think it'll move into standard of care. But we have to wait and see. And of course, you know, we're very excited, even more excited about the combination with these novel therapies, because we think that they can potentiate many of the therapies. And really, the idea is the cancer. This is what I talked about in my presentation this morning. The mistake is being to just go after the magic bullet. So try to get something that kills only the bad cells and not the good cells. And of course, you know, we're getting closer and closer, and if immunotherapy is starting to work, but it usually works in a percentage of patients, and then even a percentage of patients get resistant. And so the point, and we wrote an article recently in Cancer Discovery discussing this, is that we probably need the combined therapy, meaning the fasting mimicking diet is wide-acting, is really revolutionizing the environment for the cancer cells. So it's affecting the growth factors, the glucose levels, the ketone bodies, the fatty acids, on and on and on, the angiogenesis, everything is going to be changed. And the cancer doesn't like that. And that's when the targeted therapy comes in, whether it's immunotherapy or kinase inhibitor or chemotherapy or radiotherapy, and finishes the job. So I really envision the community, molecular community, and I come from that community, is very resistant, because we have this idea that it has to be always specific and targeted and you know, molecularly very precise. But that's a mistake, you know, because if you only do that, then you're just fighting a very difficult fight one at a time. It works for you, but it doesn't work for you. And what I do with you, And then you change and change and change. And by the time you get to the end of it, plus there is very little effort put into protecting the normal cells, right? So it's a very bad combination when you don't protect the normal cells and you keep changing therapies, hoping to get one that works, because now you're doing tremendous damage to the patient. So now the FMD does both. Protects the patient. I mean, of course, we have to still demonstrate, but we already have 3 trials that published that already suggesting that. So protect the patient on one side and make it and potentiate the effect of the targeted therapy on the other side. I mean, clearly in animals, or very clearly in animals. Let's see what happens in humans, but I'm optimistic.
In addition to cancer, has FMD diet shown efficacy with other diseases?
Yes. So we finished a clinical trial and published it in Science Translational Medicine a few months ago on 100 subjects. And these were either normal or pre-diabetic, and so pre-disease state. Also, people that were predisposed had high inflammatory markers, predisposed to cardiovascular disease. And in the diet, 3 cycles of the FMD worked very well in reducing fasting glucose, reducing C-reactive protein, reducing triglyceride, cholesterol, reducing IGF-1, which is associated with cancer by multiple epidemiological studies. So, and the incredible thing, I think, it was that also reduced visceral fat without affecting lean body mass. And the remarkable thing is that it seemed to, as we have expected, affect the people that have a problem, but not the people that don't have a problem. So, for example, if you look at calorie restriction, if you just chronically calorie restrict somebody, all these markers go down, but they go down in everybody. So you could start, let's say, having a very low fasting glucose of 75, right, very low, and it'll drive it down even further, and you don't want that, right? That's probably where the side effects come in with the calorie restriction. In our trial, if somebody had 85, already 85, very good blood glucose, fasting glucose, it stayed there. It might have gone to 83, that's it. But if somebody had 109, it came down to 95, or on average. And so, as we suspected from the animal studies, where we could show multi-system regeneration. So basically in the animal studies, we showed that the fasting mimicking diet kills damaged cells, turns on stem cells, and then in the refeeding, and only in the refeeding, the stem cells will give rise to new differentiated cells, so new functional cells. And this is the cellular level. And then we also suspected the intracellular level, meaning the autophagy, breaking down the intracellular component and rebuilding them is also occurring in the same way. So now we're seeing this. We suspect that that's why we have this differential effect between healthy people and sick people, or people that are going to be sick. And so now we started applying to multiple sclerosis. We had a preliminary trial with 45 patients that looks very promising. We did a single cycle of the fasting mimicking diet. This was 1 week long, and the patients already reported improved quality of life. Now we have a multi-center trial in Italy with 10 different centers, and this is all in combination with the standard of care drugs. So these are going to be 5 different drugs, one of the 5 drugs, and the FMD together, and that's what we're going to test. And we're also looking at plaque levels, etc. And there is another trial on diabetes in Holland. There is a trial that we're going to start at USC on Crohn's disease. We just got funded for a trial on Alzheimer's disease in Italy. So I think we really probably going to have 20 trials in the next couple of years ongoing, and we'll see. I mean, even if it works on a percentage of these, I think it's still very good news. Yeah.
So you had mentioned that you had spoken earlier, and a little bit about what you had talked about. What do you think is the single most important message that you delivered to the few thousand physicians that were in the audience?
I think that the message is treat aging, but treat aging in a smart, molecular, evolutionary way. Don't do what a lot of people do, which is they make the mistake. For example, so many people talk about 16 hours of fasting, 18 hours of fasting. Well, what they don't know is there are multiple trials or epidemiological studies showing that this increases gallstone formation, gallstone surgery, if the 16 hours include skipping breakfast, which is almost a must. This also is associated with increased cardiovascular disease, not improvements, but worsening of mortality and cardiovascular disease. So, this happens all the time. I call it, in the book, I call it one pillar strategy. You look at a clinical trial and you go with that. You look at nothing else. In the book, instead, I didn't talk about it this morning, but I extensively talk about it in the book, 5 pillars. So epidemiological studies, clinical studies, basic research, and studies of centenarians and studies of complex systems. And for example, centenarians, I feel it's a really important. It's why I follow all these people that are very old from all over the world, because in the lab we come up with something and we get excited, and then you might even do it in clinical trials or epidemiological studies and match. I'd say great. Then I go to the centenarians and say, how close is it to what they've always done? And if it's not, a word, because I think, what if you do this for 20 years and then it turns out to be a disaster? Maybe it helps you lose weight or whatever else, and then it's a disaster later on. So I think that's the important message. Treat aging in a sophisticated way, in a 5-pillar based way, and then the second message, of course, do it with the fasting mimicking diet that is periodic, that really matches all these pillars. And it was developed with those pillars in mind. Epidemiology, the centenarians, they do fasting all the time. We looked at clinics that have been doing long fasting and just asked them, did anybody ever have problems? So we really did the homework for history, and then, of course, the clinical studies confirming, and the mouse studies. Now you have all the pillars. And you feel pretty good about. And I also tell people, don't do the fasting mimicking diet, don't do the prolonged diet unless you need to. So you don't need to do it 12 times a year unless you're obese with multiple risk factors for diseases. The average person is going to do it once every 4 months, so 5 days every 4 months. That's all you need to do.
This is for overall longevity and wellness, you're saying?
Longevity and wellness. Let's say you have a little bit of high cholesterol, 210. That's okay. Do it once every 3 or 4 months.
And you're still showing great efficacy with that.
Well, we showed that in the 3-cycle trial, then we waited 3 months, and 3 months later, 60% of the effects were still there.
Wow.
So you could tell it was starting to go away, but it was long-lasting, right? So we suspect that, of course, the doctor is going to make this decision, but somebody has a 210 cholesterol, you can put them on the fasting mimicking diet every couple of months, or you can do 3 cycles. Hopefully, the cholesterol, like it did in the trial, goes down. And then you say, okay, now you go once every 4 months, and let's see what happens. If they come back and cholesterol is up again, and maybe they need to do every 3 months or 2 months. So I think it can be adjusted based on the need of the patient.
So you have this new book coming out. Will you speak to that and the title?
Yeah, so the title is The Longevity Diet. And this was the #1 bestseller in Italy in the nonfiction categories for 2016 and hopefully 2017. So it did extremely well. It sold almost, I think, 500,000 copies in Italy alone, and now it's in a few other countries. But the next one is the U.S., and January 2nd is being released in the U.S. And the book talks about my journey in all of this. So
is it geared towards the layperson or a clinician?
Layperson and clinicians. So really in Italy, one of my #1 client was doctors and nutritionists.
Wow.
I mean, a lot of I heard from hundreds and hundreds and hundreds that they really felt, because I cite everything and I discuss it from a molecular perspective, but also from a medical perspective. I talk about clinical trials, and so I think the doctors really appreciate it. It was also straightforward, but the doctors can go pick up the citations and read them. So I think that's why they really liked it. Of course, the layperson is not going to care about the publications, but they can just read the book. So half of it is about aging in an everyday diet. So what do people that live a long time around the world, whether it's Okinawa, Loma Linda in California or Sardinia, etc. What do they eat? But what part of this matches the mouse studies and the epidemiological studies, etc. So you put it all together and you have an everyday diet. And then the second part is FMD, the periodic fasting mimicking diet, and then diseases, right? What about diabetes? What about autoimmunities? What about neurodegenerative diseases? And what about cancer? So these are the main chapters in the book. And I try to, always tell the audience to be very careful. If you have a disease, best to do it as part of a clinical trial. And if you cannot wait for the clinical trial, for example, cancer, but also other diseases, I know there are people that came to me desperate and say, I can't wait another 5 years for you to finish the clinical trial because I'll probably be dead. Or even better or worse, my doctor told me I'm going to be dead in 6 months and they want me to wait for the clinical trial. So, in that case, I think we basically say, okay, let's work with your oncologist or let's work with your endocrinologist and say, obviously, or autoimmunities. There was another category where people were like, I cannot wait 5 years. And so that worked pretty well, and I think if it's done carefully, we basically, I stress the informed consent. You need to tell the patient that we don't know how this is going to go. It could go good, it could go bad, it could go worse. So I think that process is very important, and that's what we try to do when the disease is involved. Everybody else, no problem, but for people that have diseases, when you're entering this area of a clinical trial with one patient, you need to sort of have the same rules as the clinical trial and really inform the patient. You realize you're now waiting and you're taking a risk and go.
In regards to the neurological diseases and autoimmune diseases, et cetera, that you've shown efficacy with, are you showing slowing of progression or are you showing reversal?
Well, the neurological diseases, thus far, we publish only mouse data, and we're showing protection in the Alzheimer's mice, these triple transgenic mice. With people, we've done some studies on cognitive, on normal subject, non-Alzheimer's. But we haven't published that yet. It's very promising.
Wow.
But we're starting the Alzheimer trial. We just got funding from the Italian Ministry of Health for an Alzheimer trial. So, we gotta wait and do it.
So how have you implemented all of this into your own personal life?
I do the fasting mimicking diet twice a year. I think most people in my lab do it, and I don't tell them to do it, but they do it on their own. With
and they're doing the Prolon, or they're doing
no, they do it with Prolon.
Okay.
We made the mistake of in my Italian book, I made the mistake of saying, you could do it with Prolon, or you can find an expert to do it, and B was a disaster. So this is why. A lot of complaints, a lot of problems, and I said, that's it. We can't bypass the system. There's a FDA system, but there's also a process that not necessarily FDA approved, but where you do clinical trials and randomized, and you have to respect the fact that if it wasn't tested clinically in exactly the way that you're recommending it to patients, now you're exposing them to some type of danger. And in this case, there's a dual possibility of damage. One, does the doctor really know how to make a fasting mimicking diet? And few do, And that's a complicated thing. The box of Prolon contains like over 60 components. And two, if the doctor tells somebody recipes, what is that person going to do at home? Are they going to understand,
right?
Or are they going to understand what it was just told to them? Or are they going to make a mistake? Did they write it wrong? Did they go buy the wrong ingredients? So that is just too many chances for disaster.
I do want to note, though, that you receive no monetary benefit from the sales of Prolon, correct? You put it all back into research.
Yeah. All my shares will be donated, already assigned to my foundation. It's called Create Cures, and 100% of my shares. I get no consulting. I get nothing. So, I don't benefit in any way, shape, or form from Prolon. But I think it's. My good intention was like, okay, well, maybe a good doctor can recommend it the right way, but it just didn't work like that, And maybe, of course, some of them did, right? I'm not saying that all doctors couldn't come up with their own fasting mimicking diet. We published on it. We were, we had information about how it's done, and we didn't hide anything. But it's just much more difficult than people think to make sure that this is done with the right components in the right way.
And the most important question is, can you find a good Italian meal on this side of the Atlantic Ocean?
I think so. You can find very good places in Los Angeles. I mean, Vegas, I'm not that familiar with, but I imagine there's going to be some good restaurants in Vegas too. Yeah. I see some of the big Italian chefs' names on the walls around here, so I'm assuming their restaurants do pretty good. Yeah.
I want to thank you for your time, and best of luck with the book, Longevity Diet.
The Longevity Diet.
We'll look forward to reading that. And thank you for all of your efforts and for just putting this out there, and for all the people that you will be saving and curing and keeping young.
Yeah. Well, thank you very much, and glad I could participate. Yeah.